Okay, so I wanted to continue on with the plant tissue culture idea, and in a couple of weeks you'll be taking a stab at this in lab. And so there's I just posted a short video. I think it's about 8 minutes long in the reading videos section of Educat on this topic. And so if you could watch that, and he goes through the general process of how to do it. But realize that there's more than one way to do it. Like he's showing how to do it from stem cuttings, like taking stem tissue and zooming into the node region of the stem where we have, meristematic tissue, because the node regions have the axillary bud area, the meristematic region. And so that's a common strategy, is to take that region of the stem tissue. But you can take pretty much any area of the plant that's living. And I've often done leaf tissue. I've done the carrot taproot idea. That works really well. I've even taken like the root cuttings. So I think in lab, what we're gonna, if we're gonna work with tomato, and we're gonna provide you with sterile tomato seedling, and you'll be able to try different sections of the plant to try to clone it. And so we'll probably ask you to take some leaf sections, the apical meristem, we'll probably ask for that, and then the root apical meristem as well. And so we'll see what works better. Hopefully we'll have enough tissue for that. So there are a lot of different strategies out there. If you go onto the internet, you'll find a lot of resources and how-tos, and some differences of opinion on how to do it. But one of the big things is that you want to try to maintain sterile conditions, of course. This is such an artificial system that if you get a microbe in there, often they just overwhelm the system, and the plant will die out. So here you're looking at, we saw this last time, but pointing out on the jars here, they have them labeled. And so very often you do this in stages, where there's the so-called initiation stage on the far left here. And then you can see the goal is, especially for commercial production, you want to multiply it. So you want to get many of these going, and then get them to maturity. So they're talking about getting it, the elongation step is really kind of getting them more sturdy and grown up, and then the rooting phase. So all of this process, and again, you can do this slightly differently. Like this person has a step-by-step. Sometimes you can shortcut steps and do it all in one fell swoop. So it just depends on, a lot of it, what it depends on is the media that you're using and the recipe. And so the beauty of plants is that this totipotency concept, you can pretty much take any plant tissue as long as it's living, and if you know what you're doing, you can clone that plant and create more plantlets for commercial production if you want. And so and there's many different plants that are routinely grown this way, especially a lot of houseplants. That video that I posted, he talks a little bit about that there's many common houseplants that are done this way. And so I think we left off around here, or maybe the next slide, but a couple of things I wanted to talk about is this idea of working in sterile conditions, using sterile nutrient media. So we're going to be providing that all for you. And so the culture vessels that you're going to have that have the tissue culture medium inside, all of that sterile and ready to go. The tomato seedlings will be sterile and ready to go. They'll probably work in my research lab because I have 2 flow hoods in there that provide a sterile work environment. And so one of the biggest things to worry about is this idea of sterile technique. And so when you go into lab, you're going to be trained on do's and don'ts on sterile technique. And so this is something that is, you're kind of the biggest culprit of if things go wrong. It's often something you did or something that fell off your body or your arms that fell into the tissue culture medium, and then it had a chance to overgrow the system. And so sterile technique is just, they often talk about aseptic technique. You might hear that phrase as well. Aseptic just means without contamination. And so when you work in the area of plant tissue culture, or you work in a microbiology lab in general, you tend to get very paranoid and vigilant about this idea of aseptic technique or sterile technique. And so when you will be doing this experiment, you'll be asked to be very careful about how you're positioning your work environment, all your tools, the plant. You'll be asked to move your hands in a certain way. You can't be passing your hands over any tissue or over any open containers, because again, you're the biggest source typically of contamination. And so the goal is, of course, to be very careful and mindful of how you run the experiment so you're not accidentally introducing contaminants into the culture vessel. And so that is a big hurdle, and often things can go wrong in that process. But if you do it often enough, you start to learn the tips and tricks and what to do and what not to do. The other aspect of aseptic conditions that you don't have as much control over is the idea of endophytes that are in the plant tissue. And so we've talked a little bit about this already, but I just wanted to stress that you have to be aware that plants don't live without microbial partners. And so any plant tissue that you take and work with in tissue culture, there's likely endophytes that are lurking inside. Is that is that going to cause your tissue culture experiment to fail? Not necessarily. And so it really does depend. There are ways to try to minimize the endophyte gaining control or overwhelming the system. But, when you look at the endophytes, it's, fungi and bacteria that we're thinking about, and they can often come out of the tissue that you put onto the growth medium. And you can usually tell that it's an endophyte that's coming out of the tissue versus something that might have dropped into your vessel from your body or your hands or your hair. And so sometimes you can introduce into the tissue culture medium antibiotics to try to deal with that. Again, though, I've had not very good success with that, because often the antibiotics that I put into the tissue culture medium, the plant tissue didn't like it either. And so I've actually killed, like, massive kill off of the plant tissue, because even though the company said, oh yeah, you can do that. Then I called them back and I said, all my tissue died. Oh yeah, some plants are sensitive to that antibiotic. So you got to do a little bit of research ahead of time. So I tend not to put in antibiotics and kind of cross my fingers and hope for the best. I've also seen the situation where endophytes start to grow out of the plant tissue onto the tissue culture medium, and they don't overwhelm the system. They're there, you can see them, but the plant looks happy. The microbe is staying in check, so to speak. It's not, growing over everything and attacking the plant. And so I've seen all kinds of situations where the endophytes are there, and sometimes they cause problems and sometimes they don't. There's no way to serve, when you look at tissue culture techniques, the idea is to take the plant matter and do surface sterilization. And that video I posted, you'll see him doing that. He pops the stem tissue into a flask of a bleach solution. And so what he's doing there is using surface sterilization techniques. And very often that's, you do some kind of bleach treatment, and then sometimes you do usually 70% ethanol treatment. I'm going to abbreviate ethanol, ETOH. That's a common abbreviation of ethanol. And so That surface sterilization process, by the way, can vary tremendously depending on what plant you're working with, what part of the plant you're working with. And so, again, if you go onto the internet and start looking, you're going to see all kinds of opinions on this and different protocols. And so, how much bleach, what percentage of bleach, how long should the tissue soak in that? And then, should you do bleach and ethanol or just one or the other? And then often no matter what method, then you tend to soak the tissue in sterile water. I'll abbreviate that H2O, sterile water at the very end to get rid of the bleach or the ethanol. And so I find the toughest part of tissue culture really is the surface sterilization. To try to pick the right recipe to have success is kind of a troubleshooting process, and you have to be either, find a really good resource that is working exactly with the plant you want to work with, or you're going to have to deal with a bit of troubleshooting. So just be aware of that if you ever do this. And there's a lot of people that are getting into this in their home where they can create their own home, like flow hoods, and you can do this at home. I had a student who took plant foods last fall with me, and this past summer he sent me pictures of his home system that he was doing tissue culture work and growing houseplants with tissue culture strategies in the basement of his house. So it's doable. You just have to invest, and it's not gonna. You can do low tech ways of creating like a flow hood, like the flow hoods you'll work with in my lab. Each one of those costs like over 10 grand. But there's other ways to do it, and actually, it can be as simple as getting a tote from Walmart and tipping it on its side, working inside the tote. And so we've done that as well and had success with it. So just be aware that there's a lot of resources out there. There's a lot of opinions on processes, but in general, what you're playing on, what you're capitalizing on is that the plant's ability to be totally potent and to grow and develop from a cluster of cells. That, and as long as you provide the right type of media, then you have a high chance of success if you can manage to not get contaminants in the system. And so we use mineral salts. Media is often how it's abbreviated, and people will refer to it, they'll call it MS media. That stands for mineral salt. So you'll hear that a lot. And those micronutrients, they're often some different minerals that are put into the media recipe. Then they usually add sugar because the plant can't do enough photosynthesis by itself initially, especially. So you provide some sugar for the plant. And then typically and again, the different hormones involved, there's a variety of hormones you can pick, but typically the hormones involved are a group of hormones called cytokinins. And often a very popular one is called BAP or BAP. That's its abbreviation. Benzoylaminopurine. We work with that one a lot. But it's a type of cytokinin, which is a plant hormone. And then the other group of hormones that often get put in are called auxins. And often you'll hear people say NAA. That's as an abbreviation for a type of auxin. Okay. And so there's synthetic versions of these auxins that are stable, they're easy to pop into this recipe. And what is so good about this is that these hormones will stimulate not just cell division, so the plant is growing, it also is encouraging the plant to differentiate, because of course you have to have a total plant. You just can't have a mass of cells. You have to have root tissue, stem tissue, leaf tissue. And so these hormones in the right ratio, all right, so sometimes you'll see them all mixed in at the start, and there will be a defined ratio of these two hormones. Sometimes you'll create several different recipes and transfer the plant into these different recipes with different hormone concentrations to kind of do a step-by-step process. So it just depends. There's different strategies. But basically, you definitely need some kind of hormones in there to encourage root growth at the right time, and shoot and leaf growth, of course. And so, um, and then organic complexes are often added, and just for an extra boost of growth factors that the plant might um, find, it's good nutrition for them. So coconut milk, it's actually endosperm. When you think about coconut milk, it's actually endosperm of the coconut fruit. And sometimes yeast extract is put in. So you can buy like all set, ready to go mineral salts media from companies out there. It's all like packaged into one bottle. And so it's getting much easier. You don't have to do it from scratch necessarily. And so um, sterility and good technique when you're working with the tissue is really critical. But we work with these types of vessels, so that's what you're going to work with in lab. These magenta vessels are called, and they allow for, they're tall enough so that the plant can get room to grow, but it provides a sterile container. And the trick is, of course, is to make sure that you're not introducing any kind of microbes in there that can overwhelm the system. And so the tissue culture is kind of the standard phrase, but I'm seeing more and more micropropagation. They're getting fancier about it. So they're using that term a lot. And so you might end up seeing that, like if you get a job in a company where they do tissue culture, you might refer to it more as micropropagation. One of our former students here is downstate working for a cannabis company, and he's largely doing a lot of tissue culture work. And then one of my other students who graduated a few years ago, she's downstate working for a different cannabis company, and she texted me during the summer. She said she wanted to reach out to Ben, who's doing a lot of tissue culture work at that other company, because I don't know if you heard, but there's a disease sweeping through cannabis. I think it's a fungal disease. I'm blanking on the name of it at the moment. Does anyone know? It started out in California, but it's sweeping across the country. Has anyone heard about this yet? I'm blanking on the name of it. But anyways, they are super worried about it. Downstate, they're noticing that their plants are being powdery mildew. It might have been powdery mildew. Yeah, but it sounded like it was something like something new is popping, like a new strain or something. I don't know. HLV. I think that was it. Yep, that was it. And so she was reaching out. She wanted to reach out to Ben to get new tissue that doesn't have the virus in it. And so they were like scrambling to find a way to grow cannabis with a high chance of the virus not being present. Tissue culture can be one way to do that, especially if you take apical meristem tissue. For some reason, and I don't know, I've tried to chase this information down, but for some reason, the apical meristem region of the plant, viruses have a hard time infecting that particular region. And so you often see, you'll hear or read resources where they say, it's best to take apical meristem tissue if you want disease-free tissue. And so obviously, if you're trying to mass produce thousands of plants for commercial production, you want disease-free. So, but I'm not exactly sure the biology behind that. I don't know why viruses seem to be, they can't quite breach into the apical meristem cells. Okay. So tissue culture is an up and coming field of in the commercial crop realm. It's really becoming heavily utilized. Many houseplants that you see for sale are grown through tissue culture, and it's a very fast and efficient way to grow plants in a very small space initially, and then gradually you do get these shifted. Like once they form good root systems, then you have to acclimate them. They use that word a lot. They'll say acclimate. And so you need to take them from those enclosed vessels, which is very humid and very moist, because the agar, that tissue culture medium, has a lot of moisture. And then you have to gradually acclimate them or get them used to drier conditions, like if you want to put them into soil in a pot in a greenhouse. That's a big shift in their environment, their parameters. And so you have to gradually do that. Like you can't just plop them, take the plant out of the culture vessel and plop them into soil and call it good. You have to know that there's a step-by-step acclimation process. So, but it's not that hard. You just have to get them used to it, and they're pretty quick at adapting. So it doesn't take that long either. And so tissue culture or micropropagation is, initially it was used a lot for scientific research, but it's really gotten out into the normal everyday world where people are using it for commercial production. So a lot of benefits to growing plants in that way. Okay, I did want to talk about some other methods though of how do you work with plants, especially if you can't have them mating through normal breeding practices in the sense of taking the pollen from this and putting it onto the stigma of that and having everything work just perfectly. So the fancy way to say that is if they're sexually incompatible. And so if you can't get these plants to breed with each other and create offspring, then an option might be to go into the lab and do this through other methods and using biotechnology. And so these are not the traditional breeding methods that we're talking about because they're sexually incompatible. So one strategy is to use this particular bacteria called Agrobacterium tumefaciens. That species I would like you to memorize, please. That is something that is used so heavily. I'd like you to be aware of its name. And so what this bacteria is really quite amazing because it's a plant pathogen. And so and the other thing too is that it infects a whole bunch of different plants. So they say it has a broad host range. It really can attack a lot of different plants very successfully. And the reason scientists got so excited about this bacteria is that they saw that it can insert its plasmid into the plant chromosome. So here's a bacteria DNA getting put into the plant DNA. And that was, when that came out, that was just like mind-blowing information that you could have that kind of shifting of genetic material from a bacteria into the plant. And so why they got so excited was not only was that such a fantastic thing to think about, but then they said, you know what, we could use agrobacterium as a tool to transfer genes into plants that we want to go into plants. You have to appreciate trying to get genes into plants without agrobacterium is tricky. Plants are not very easy to work with in the lab in the sense of trying to get genes into them because they have a really tough cell wall. There's a lot of unknowns. We just don't know as much about them in terms of how to work with them in this manner. And so when this came around, the molecular biologists that do plant research thought, we can use agrobacterium, mix agrobacterium in with some plant cells, and agrobacterium will do all the hard work for us. Just take the gene that we want. So then the goal is, okay, let's take agrobacterium. So here what you're seeing in this diagram is the purple is the agrobacterium cell. And you can see that it carries this plasmid. Plasmids, remember now, those are just small circular pieces of DNA that many bacteria have, in addition to their normal chromosome, which is over on the left. They call this the Ti plasmid. T stands for tumor inducing. The reason they call it that is that when this bacteria causes plant disease, it forms a tumor on the plant that you can typically see with the naked eye eventually. Whoops. So they call that tumor in the plant world, we call it a gall, G-A-L. And I'm sure you've seen these, just even walking around Marquette, you can find them on trees. On tree trunks, you see those, like, it looks like a tumor growing off the tree trunk stem. That's probably agrobacterium that's causing that. But just be aware, there are other organisms that can cause a plant to form a gall like that. Now, is it cancer? I've had that, students think, well, could it be cancer? Is that what's going on there? And in a way, it's kind of analogous because in the sense that that gall, that big massive tissue, I mean, it's tumor-like in the sense that it just grows and grows and grows and grows and grows, and it just forms a lump of cells that really don't do anything for the plant. It's not helping the plant. Just like cancer cells in us, when cancer grows in us, it's a tumor of cells, our own cells, but they're not really functioning for us. Like lung tumors don't help us breathe, right? They're just taking up space in the chair. And so it's kind of similar in that regard, but it doesn't cause cancer per se. Like many plants can survive agrobacterium attack just fine. And like those trees you see, especially around here, they look fine. They're green, everything looks good. And so this particular pathogen usually doesn't kill a plant. It might weaken it if it's already getting hit by other stressors, but it usually doesn't kill it. But anyway, so that's why they call it tumor inducing, because that's what it actually does if it's allowed to be pathogenic. But what the scientists do is they knock out that gene so it won't form a tumor. So they take that one out and they put their gene of interest in. So here they're showing that they're putting in some stretch of DNA that they want this plant to be able to have and express. So maybe it's for disease resistance. Maybe they're trying to introduce hops, latent virus disease resistance into cannabis. Okay. And so what they do is they pop that gene into this plasmid of the bacteria. And then what they do is they use, and you can see it right here, they use plant tissue culture techniques. So what you have to do is you have to work with plant cells. You can't just inject this into a mature plant. That doesn't work. So you have to start kind of from scratch. You have to work with typically, well, here, let me show you this picture. Typically what you do is you work with liquid cultures of plant cells. And then you mix in the bacteria, the agrobacterium, into that liquid. So the plant cells and the bacteria are in the same broth and they have a chance to physically interact, and the bacteria can infect the plant cells. So basically, when you think about it, it's like agrobacterium is just a tool or a vector to introduce the gene you want into the plant. Okay. And so they've really capitalized on its easy ability to infect. So here is, in this case, they're using the idea of herbicide resistance versus herbicide susceptible. So they're saying in this example, they're saying, what if we want to create this plant to be herbicide resistant? And so that we can spray herbicides in that field to kill off competing weeds, but our crop plant will be resistant. And so if it's initially susceptible, what you can do is using tissue culture, get some of the plant cells going, and then shift them into a liquid culture. And then in this case, they are using agrobacterium that have that altered plasmid they inserted the gene of herbicide resistance into the plasmid, pop the bacteria in there. And under the right conditions, the bacteria will actually get into these cells, into the plant cells, stick their DNA into the plant chromosome. And then what you do is, if you're trying to figure out, well, which of these cells got infected, what you do is you put the herbicide in there after a while. And then you're exposing those plant cells to the herbicide. Those that didn't get infected by agrobacterium those cells will die off because they're still sensitive. Those that persist, that agrobacterium was successful. It got the DNA in and made it resistant. And so then you take those cells, those plant cells, and you start to put them into tissue culture techniques and get them through the tissue culture process to grow into a complete plant. So when you're done, every single cell of this plant will be resistant because you started with just individual cells here of the plant that was infected. And so if all the growth is coming from those cells, you know that every subsequent cell, as it's growing, is going to have that gene for resistance. And so what you've essentially done is the whole plant body now is resistant to herbicides. Okay, and that's what you want if you're going to be spraying a plant with herbicide. And so it is just this step-by-step process. And because agrobacterium is so good at infecting plants, it's just become a wildly popular tool to introduce genes of interest that you want into the plant species you're working. And so this is done, they've tried this out with a lot of different plants, and agrobacterium, it's not 100%, agrobacterium doesn't affect every single plant out there, but many, many plants. And so it's been used widely. Here they're looking at this idea again, disease resistant in this case instead of herbicide resistant. So one thing to be aware of, you know, like here, what they're doing, so they're like, okay, we want to make this this plant, you know, disease resistant. And in order to tell whether or not it worked, you often have to tag or mark your gene of interest. And very often you use antibiotics for that. And so what you do is you put your gene of interest, disease resistant, you put an antibiotic gene right there with it. So then if the plant cell takes all that up, then the way to tell, okay, you get the disease resistance gene in, you grow those plants on tissue culture medium that has a bunch of the antibiotics. And if they survive in the presence of that antibiotic, then you know, because they have that marker, the antibiotic resistance marker. If they survive the presence of the antibiotic, the assumption is, okay, they must have taken up that whole cluster. They took up the disease resistance gene plus the antibiotic gene. And so it's just that antibiotic marker gene, that resistance gene is just there so you can tell which plant cells got transformed and which didn't. And so when we're going to do that transformation experiment in lab in a little bit, that's what we'll be using. It'll be an antibiotic you know, can they survive in the presence of the antibiotic idea? But this is just showing the agrobacterium being used as a vector, essentially a way to transfer in the gene of interest into the plant. And it's quite remarkable that plasmid DNA coming from the bacteria goes right into the plant chromosome, and then the plant will start expressing that DNA. It's really quite amazing that you have this, you know, two different kingdoms of life, and yet you have the ability of them to transfer, you know, that DNA from one kingdom to another kingdom of organism. Really quite remarkable. But again, using tissue culture techniques to determine which plant cells got successfully transformed and then growing those up into a complete plant. So you can see how tissue culture techniques can be super helpful for this kind of idea of trying to introduce traits that you're looking to do. And especially if, you know, you can't do it through normal breeding methods, this is, you're kind of limited on options. There's only a handful of things to do. Questions on this idea? The next slide is just giving you the full detail in words, so that's for study purposes. So it's part of the book as well, but I thought I'd put the actual figure legend. Okay, what else? Okay, so agrobacterium is a wonderful tool for being able to transform, genetically transform a plant. But there's other strategies that are done, and one, if you're going to do it without agrobacterium's help, then very often what you end up having to do is you need a lot of the copies of the gene that you're trying to get into the plant. You just need a lot of it. you need to make many, many, many copies because it doesn't have a real high success rate. And so one way to do that is to, again, use the help of bacteria to just make a lot of copies of this gene of interest. And so very often what they'll do is they'll get E. coli, which is, we've all heard of E. coli. That is kind of another workhorse bacteria that's used heavily for something like this because we know how to grow it, we know how to keep it happy, and it can multiply really quickly, which means all of the DNA inside of itself multiplies really quickly too. And so it's just a way to get a lot of copies of the gene. You put it into E. coli and you give everything E. coli needs to be happy, and it will just make lots of itself, including all of its DNA. And so you get lots and lots of that gene of interest through just using E. coli to grow it up for you, essentially. Okay. So that has been done for decades and can be very successful. So you put your gene of interest into E. coli, often through a plasmid again, and then just let E. coli make many copies of it as it goes through cell division, cell division, cell division. E. coli can double its population in under 20 minutes. So it doesn't take long to get many, many, many copies of your gene of interest. Another way to do it, though, if you want lots of copies, is just use PCR. PCR would work too. And that's, you don't have to fuss with E. coli. And so once PCR was discovered, a lot of people switched to that and said, ooh, this is pretty straightforward. You don't need to worry about growing up E. coli. You can just have a machine do this for you. And so again, the strategy here is to get your gene of interest. You can get many, many, many copies of it made. And that's what you need if you want to try some of these other techniques to genetically manipulate the plant without the help of agrobacterium. So basically, we're looking at this idea that you can get genes into plants through non-breeding methods. And so it's much quicker. You don't have to go through the whole breeding process to get offspring that have the genes of interest. You can do it in the lab in a fraction of the time. And then you can get genes transferred from wildly unrelated species. All right. And so they don't have to be super closely related. And so there's been, like the jellyfish green fluorescent protein has been put into a lot of organisms to get them to glow. I mean, we're talking about a jellyfish in the ocean, taking one of its genes, putting it into a bunch of land-based different organisms, including different animals too. So it happens. Okay, so if you can't use agrobacterium, but you got to get genes into the plant, how do you do it? Like I said, plants can be a little challenging because you have to deal with their cell wall. They have very tough cell walls around their cells. And so one strategy is to zap them with an electric field. And so this is called electroporation. You need a special machine in order to do this, which is super expensive. And so like we don't have one in our department. We use other methods, but other methods, for example, so the electroporation, by the way, it just weakens the cell wall enough. It kind of makes it holy, so to speak, to allow the DNA to get through, but it doesn't like totally degrade the cell wall. It just kind of like temporarily relaxes it enough to get something through it. You could also treat the cells with polyethylene glycol, and they have found that that seems to help relax the cell walls a little bit too, so that you can get DNA through that and into the cell. Gene guns, believe it or not, and it actually looks like a pistol. I worked with one when I was a grad student when a fellow student in the lab was trying to genetically transform fungi. And so fungi are also very challenging. They have cell walls on a chitin, which is, again, pretty strong. And so getting DNA to fungal cells, you actually shoot it in. You shoot it in on bullets, really, really tiny bullets. And they're usually made out of gold. And you coat the gold pellet, like tiny, tiny BBs, think like that. And they're made out of gold because gold, it won't irritate the living organism. It's pretty inert. But you coat it with DNA and literally you shoot it in. It was so fun. We were doing this in the lab, and it was just really weird to see. We rented the gene gun from a company. Couldn't buy one; it was too expensive, but they let us rent it from them, and it worked. It didn't have a huge percentage of success because he shot DNA into a lot of different samples, but he did get success with it. So it's you gotta be. Oh, and then micro injection, you can actually do a tiny um, syringes, needles, so you're working with a microscope, you're looking at these plant cells under the microscope. It's kind of like you probably might have seen video of in vitro fertilization of sperm and egg. It's kind of like that idea, except you're putting DNA instead of sperm DNA. You're putting, a gene of interest that you want. And so the idea here, whoops, this word protoplast, you'll hear that term a lot when it comes to how to insert genes into plants. So protoplast, what that is, that's just a plant cell that doesn't have a cell wall anymore. So there's no cell wall. So you can take plant cells, and I've had undergrad students do this. We thought about having a lab exercise on this for this class, but the problem is it's a several hour process, and we can't get it done in 3 hours. We would need like 2 days in a row, back to back. So that I can't really quite fit it into this lab, this class. But I had an undergrad doing research with me one summer, and we were playing around with this, and he got it to work. And so it's basically you get some enzymes they isolate these enzymes from bacteria and fungi, and you get some plant cells in a liquid culture, and then you spike in these enzymes. And if you do it right, the enzymes will attack the cell wall and get rid of the cell wall, but not mess with the cell membrane. So a protoplast is basically the plant cell, it still has an intact cell membrane on it, but no cell wall. And then you can look at that under the microscope, and you can actually see them. You can see these protoplast cells under the microscope. And so then you would take your fine needle and inject your DNA of interest into that, like in vitro fertilization. And that can work quite well. But again, you have to know how to generate protoplasts successfully that stay alive, and then use microsurgery, essentially. Liposomes, this is the idea of encapsulating your gene into a lipid like balloon, so to speak. It's just you mix your gene of interest with some lipids. So what you're trying to create is a phospholipid bilayer with the gene inside of it. So it creates like a bubble of this lipid. And if you look inside, your gene's in there. And then what you do is you take your liposomes and you mix them in with protoplasts. So you have this liquid culture, you have your protoplast floating around in this liquid culture, then you have your gene put into these lipid vesicles. The fancy word is vesicle. It's just like this little tiny balloon of lipids, the gene inside. You pop those in with the protoplast, and they'll fuse. The lipids, the vesicle, the lipid vesicle, that membrane material will fuse with the protoplast, and that allows for the gene to kind of get dumped into the protoplast. It's really quite amazing. It's kind of similar to how viruses get into you, how they can sneak in through getting encapsulated by lipid material and get into you that way. So these are different techniques where if agrobacterium isn't going to be the route that you can go, there are, these kind of techniques. This is the gene gun idea, where you have the plant cells. Usually you grow them on an agar plate like that. When we did it with fungi, it was fungi growing on an agar surface, and then you just bombard them with bullets that have the DNA on there. Kind of fun. And then here is the microinjection idea. Those are protoplasts that you're looking at. But all of this is looking at the idea of if you want a particular gene to get into your plant of interest and you can't do it through breeding methods or it's too slow through breeding methods, this is another option and can be very quick, much faster than normal breeding methods. So this diagram is just trying to show some of the different strategies employed. This is kind of like a big picture idea of options that you can do in order to get an entire plant with your gene of interest through the entire plant body. So they can work with all kinds of different parts of the plant. They've even worked with like pollen grains and bits and pieces of the flower. They've done that too. So. But again, this is, commercially what a lot of this is being used for is like herbicide resistant. They can use genetic engineering so that the plant you're trying to grow will be resistant, but if you spray your field, all the weeds around in the field will die off the herbicide. And so this was a huge shift in agricultural strategies when it came about. And you can see on the right, there's, all those weeds. If you're trying to grow corn, you don't want all those other plants. By the way, the definition of a weed, I remember when I was a postdoc at the University of Minnesota, they have a whole department called weed science, not cannabis weed, but just like weeds in your field, weed science. And so I remember talking to one of the postdocs, and I'm like, so what is a definition of a weed? How do you define that? And he laughed and he said, a weed is a plant growing somewhere where you don't want it to grow. That's it. So corn could be a weed if you're trying to grow a bunch of alfalfa and corn pops up. And you're like, I don't want corn, I want alfalfa, And so it's kind of interesting that they have a whole department on, weed science, but it's really kind of tricky to define. There's no like exact weed. It's just a plant growing where you don't want it to grow. A virus-resistant papayas, they've used that genetic engineering for that kind of idea as well. They also use genetic engineering to help plants deal with soil that is not very hospitable. So maybe it's toxic metals in there or too much sodium or lacking in. So there's all kinds of options that I've seen published on how they've engineered the plant to grow in soils that aren't ideal. And yet the plant is able to survive. This one is looking at salt, high levels of salt. And they're showing that you can see in E how the top plants, the pot with E in it, had the highest amount of salt. And then those are the control pots up top. And then on the bottom are the ones that were genetically engineered to deal with salt. And you can see the highest concentration is off to the right of salt. And you can see they're all quite happy, even at very high concentrations of salt, because they had those genetic modifications done. Flavor Saver tomatoes that have been on the market have also been done through genetic modification. Okay, I have a few other things to say about biotechnology next time, and then I'll probably talk a little bit about propagation as well, because it kind of goes hand in hand.